Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 151st seminar, and I call it a DTU day. For almost 6 weeks, since I returned from the SynBYSS conference in Hawaii, I have not traveled at all except for giving an invited talk at UC San Diego, which is literally located within walking distance. However, in 10 days, I will embark on 4 weeks of back-to-back trips to Spain and Portugal, as well as New Zealand. Interestingly, today's speakers are both neighbors to those countries, one from Denmark and the other from Australia. What a coincidence. Also, originally, both of them are from Germany, but now, working in different other country right now. Also, I will visit Denmark and Australia later this year. And also, given this kind of interesting situation, now we have the speaker from Denmark and Australia. I believe this seminar really, global, becoming the global seminar or global webinar series, because of all the people participating from all over the world. So thank you so much for everybody. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Tilmann Weber. He is a professor at Technical University of Denmark, or DTU, and an associate scientific director of the Natural Products Genome Mining Group at the Novo Nordisk Foundation Center for Biosustainability at DTU. I have known him for a while and met him multiple times, including during my seminar visit at DTU. And as you know, he's of course very famous. He is also a pioneer in genome mining software development and engineering and understanding biosynthetic pathways for natural product. Kindly, Tilmann also accepted my keynote invitation at the Hawaii conference, giving an excellent talk during that conference. It is great to see him again, and also looking forward to see him again in Denmark during the Metabolic Engineering Conference in the summer. Tilmann, thanks so much for your great contribution to the scientific community and also education, and the virtual podium is all yours now. And thank you again.
Thank you so much for the kind invitation. This was one of the invitations where I didn't hesitate long, because the Hawaii trip was fantastic, but very, very long for me. Now the online seminar, I even can sit in my living room and give it. So thanks so much for this opportunity to talk to the next generation of the specialists that have to address world's greatest challenges. And I'm also really looking forward to Anna's talk later, which will be on a little bit different topic than I will present you. But I think taking all these approaches together, we really can make an impact. And now let me just try to share my screen. Can you see the screen?
Yes.
What I want to show you in the next 5 minutes is just a very brief crash course where I want to promote that microbial natural products actually are super interesting molecules that have a huge impact as future drugs, as compounds for sustainable agriculture, as compounds to protect our foods, and also are fantastic targets for synthetic biology approaches, where I think there are a lot of possibilities where we can use these technologies. So I called my presentation the molecular treasure hunt, because this nicely summarizes what my lab and my group at the Center for Biosustainability actually is interested in. We want to find and apply new bioactive molecules from bacteria for being drug precursors, and especially also for new sustainable applications in agriculture, which is one of the key focus points of our recent work. And within our group, we cover a very large area, which also directly indicates we are a very interdisciplinary team, employing chemists that really look into the molecules. And then we're using a lot of computational biology approaches based on normally genome sequencing and also other omics data sets to study the production of these compounds and then combine it with metabolic engineering and synthetic biology strategies to actually access these organisms, because we have the challenge that especially E. coli, which is a fantastic host for molecular biology, a fantastic model organism, is really not the best one to make natural products. So we have to work in the original producers and have to reinvent the wheel very, very often just to do basic genetic engineering. And I want to give you an overview, very quick overview of these aspects. You already mentioned the computational work. So we were not the very first, but one of the first groups that developed software and databases to use genomic data to study this kind of natural product biosynthetic pathways. And over the years, we have now a very nice portfolio. And what I want to emphasize very much, a lot of these tools weren't just developed in my group, but I'm very happy to have fantastic collaborators all over the world that greatly contributed to this. And some of the software tools you may actually know, and they wouldn't be there where they are without the contributions of many, many people or some of the databases too. Our main project started as PhD topics from Marnix Medema and Kai Blin, who now all have become group leaders themselves. And Kai still works as a senior researcher in my group, and Marnix now has his own group in Wageningen University. And we're still it's now getting 15 years are collaborating in developing the software, where we want to provide a platform for especially lab biologists and chemists to mine their genomic data without requiring a bioinformatics degree or a degree with data analysis. So this anti-SMASH software is a very simple and very easy to use tool where you upload your genome sequence of fungi or bacteria, and then we can extract as much information as possible out of the sequence data you provide to analyze these biosynthetic gene clusters. And you can do this for individual genomes. Recently, we have made an extension which we call BGC Flow, which you can use for very large-scale data analysis. And it's in the end only restricted by the computing power and the memory and the disk space you have available. So you can use it with thousands or 10,000s of genomes to make a systematic analysis for these biosynthetic pathways for the natural products by integrating not only our tools, but basically most of the tools that are around to study these effects. We also, together with many fantastic friends and colleagues, curate and run several databases in the field, from very small one. This MyBig database is a database that contains experimentally confirmed biosynthetic gene clusters, and it's only around 3,000 entries. And this is a large community effort. We just released version 4 of the database with more than 250 international collaborators that joined online anothathons and provided, in the end, the data sets within the database. We have the anti-SMASH database and run by Marnix's team and Nadine Zima teams. We're also involved in the very large scale, more metagenome driven databases. Now, this, of course, is very nice. You can make nice analyses, but if we really want to make an impact, it's not only that we can look in the genomes to find these pathways, we actually need to find compounds and test the compounds that make an impact. And one big source that we're using are actinomycetes, and we have our own strain collection, which is relatively small, only roughly 1,700, 1,800 strains. But what's very special about this strain collection is that large parts are completely sequenced with a very, very high quality. So when we submitted our data set to NCBI, at the time we submitted, we more than doubled the amount of high quality Streptomyces sequences that were available at NCBI. And this is now basically our treasure, but the treasure is publicly available, where we can mine for specific classes of natural products. We now can do very complex analysis, not only on single strains, but on whole family of strains. So here you see, for example, if we map gene cluster families on the location in the chromosomes, which you only can do if you have this very high quality finished sequence data. And what can we make of this actually is new molecules. And as briefly mentioned in the beginning, these actinomycetes, they are very well-studied bacteria because they are among the most important producers. A lot of the antibiotics that we use in clinics come from these, but they're also a little bit nasty if you want to do metabolic engineering with them. So with the introduction of the CRISPR methods, this really was a game changer for us. Our lab at DTU was among the first using CRISPR technologies for actinomycetes. And we now offer a toolkit with many different tools, even more than are on this single slide, where we can now go in much higher throughput than before. We're still far away from model organisms, and I'm really envy Anna for her organism because ours grow very, very slow. But still, with these methods, it's now possible to use metabolic engineering. Also, if you want to use these, all the plasmids are at Edgene, so you can just get them very easily. And finally, I just want to prove that this is not only theory, you can actually use this to find new molecules. So again, in collaboration with several fantastic groups. Here's just the overview of some of the molecules that we were working on in the last years. I don't have time to really introduce you to these. I'll just present you one, which I find mind-blowing. The molecule was found in a collaboration project with the Medina Foundation in Spain. At the time we found it, I think it was the largest bacterial polyketide. Now Brett Moore's group has found some microalgal ones that are even larger, but it's a tremendously big and complex natural product. And for the pathway, actually, you don't even need a software. You can just look at the open reading frames in the genome because it's synthesized by a very big enzymatic assembly line. It's over 178 kilobases in size. For a bacterial gene, this is tremendous 10 subunits with almost 60,000 amino acids as a monomer and 128 enzymatic active sites that all do a specific reaction. So by just looking at the sequence data, you can draw the biosynthetic pathways. And there are two small positions where our predictions are wrong. And they even made the chemists able to do a full stereochemistry assignment because for some of the enzyme domains we can predict stereochemistry. So this allowed the NMR experts to assign the stereochemistry. So I hope I could show you this is a fantastic field to employ metabolic engineering and synthetic biology technologies. We're still by far not there. There is a huge potential to improve these methods and make these kind of molecules more accessible. But integrating all our knowledge, I think, it's a fantastic time to be in this field. And I hope I could convince you of this. And before I hand over to Anna or Taesok to introduce her, I just want to thank, this is a very interdisciplinary approach. So I have a fantastic team at DTU helping me with this kind of research. I want to thank all the agencies that give us funding to do all of this, and also many international collaboration partners. I can't even mention the anti-SMASH or MIPI collaborators. This would be several slides, but here are just the ones, basically the main collaborators where we work very closely with. So thanks a lot for this. And with this, thanks, Taesok, for giving me the opportunity to share our approaches.
Thank you. Thank you. This is fantastic. You know what? I started my research career as a master student, 1998 or 1997, depending on, how you count. At the time, I started with the polyketide synthase, and then my goal was the swapping the gene. But at the time, we don't have anti-smash. We didn't have much tools in terms of engineering the bacteria. It was nightmare for me as a master student. I suffer a lot, because we manually kind of looking at the sequence and, manually kind of, do all the manipulation without CRISPR. And then I'm so glad, we see last, a little bit less than 30 years, we have the progress, a lot of progress. Now we have, more engineering than art, in terms of the natural product synthesis and engineering. So thank you so much for this wonderful talk and then, work that changed a lot in the field.
Thank you.
Okay, so now the main speaker of today with a longer introduction. Anna Faber completed his bachelor degree and master degree and also teacher training in biology and chemistry at the Philipps University, Marburg in Germany. From there, Anna moved to Perth, I think Australia, where she is currently undertaking her PhD program at the University of Western Australia. As a forest research foundation scholar, she is pursuing her PhD with a focus on combating very important problem, of course, the plastic pollution. She integrates synthetic biology, biochemistry, and science communication to drive meaningful change, uh, both in the lab and also within her local community, including Western Australia. I think I met her at a conference where I was impressed by her presentation, although she is still a graduate student. So I invite her to, apply or nominate her, becoming the Rising Star speaker. And coincidentally, after accepting her nomination as a Rising Star speaker, I got to know her advisor is also someone I know, Dr. Pritz, who is actually my Global Center Circle member. And I'm going to say what a small world or a coincidence again. With Tilmann, as I mentioned earlier, from Europe and Anna from Australia or Oceania, this webinar is truly a global event. Anna, thanks so much for your time today, especially at a very inconvenient nighttime. I mean, the midnight, right now, actually past midnight. And thank you so much again. And please take it away. It's all yours now.
Thank you, Taesuk, so much for inviting me. I'm just going to start sharing. And thank you, Tilmann, for setting the stage. That was a fantastic insight. And I'm looking forward to learning more about your research, especially at the conference. All right. So I'm just going into presenting mode, and then we will take it away. Perfect. Yeah, thank you very much for having me tonight. It's past midnight, but I'm very enthusiastic and I feel very honored for this invitation. So thank you for having me. I'm going to be talking about my PhD project, which is focusing on developing toolkits in Vibrio natriegens and then using them to tackle one of the world's biggest problems, namely plastic pollution. And like Taesuk already mentioned, I started my academic journey in Germany, but then moved to Australia, to the University of Western Australia for my PhD, and I had the great privilege to join George Fritz in the Fritz Lab here. I'm also very thankful for being part of the Forest Research Foundation, which is a international and global knowledge and research hub and brings together people from all different disciplines, and we learn, study, and research together. I also want to acknowledge that we're actually doing our research on the Wudja, the country of the Whadjuk people of the Noongar Nation, the indigenous local people on the land that the university is situated on and where I take my samples. And I want to acknowledge that those people are and were the first that learned from country. And so I see them as the first researchers of Western Australia. And I pay my respects to past, future and current elders. So, as I mentioned, Perth is a beautiful, beautiful city. It's situated at the Darrawal-Urrugun, which is the Swan River. And we enjoy this fantastic place every day. But at the same time, if you look closely, we can actually see that we also share one of the biggest environmental crises of today, which is plastic pollution. So this motivated us at the University of Western Australia to use our background in synthetic biology and biotechnology to tackle and to help find solutions for this problem. As researchers, I think we're all very fascinated, how we can use our knowledge to have a better impact on the world, and especially when you see how much plastic there is around, up to 60,000 pieces per square kilometer in the Perth CBD. You just really want to do your best. And yeah, I always start with a definition in plastics because I think it's very important to scientifically understand when we're talking about plastics. So plastics is an artificial material made from long polymers, which is shaped by flow and process into finished products. And bigger definitions also include elastomers like rubber, highly edited natural polymers such as cellophane, and then also materials with a high additive content such as silicones and PVC. The 7 most abundant or mostly made petroleum-based polymers are polyethylene, polystyrol, polypropylene, and polyvinyl chloride, which feature only carbon and hydrogen in their backbone, and then also polyurethane, polyamide, polyethylene, which also have heteroatoms such as nitrogen, oxygen in their polymer backbone. And these are the 7 most abundant petroleum-based plastics that we produce today. When we look at the recycling rate, which is one potential solution how to mitigate the plastic pollution crisis, we actually can see that the world average recycling rate is only 9%. The best countries in the world, which are actually in the EU, they only recycle 14% of their general plastic waste, and Oceania, including Australia, is doing less than the world average with only 7%. So there's huge potential to mitigate the plastic pollution crisis by developing better recycling technologies. And if you look into what is currently being done, is mainly mechanical recycling, especially for PET and PE, which mainly means that plastic waste is collected and sorting and washing occurs, and then the plastic is just mechanically grinded down, melted, and shaped into new products. And we can only do this a couple of times because the quality of the finished product is so poor. So chemists have been genius in the last couple of years, and they came up with chemical recycling, which literally takes chemical catalysts and breaks down the plastic polymers into the constituent monomers. And these monomers can then be used for new synthesis. And the product quality is much, much higher for this process, but we still need quite high temperatures, always or most of the time more than 100 degrees Celsius. And those chemical catalysts can be quite nasty. So the microbiologists or biologists in general, they came up with the idea of looking into nature and potentially finding biology, so finding life that can do the same trick in maybe milder conditions. And this is what we call microbial recycling, where we still use a catalyst to break up the plastic polymer into its monomers, but we do it enzymatically. So we use a biological protein catalyst. And about 10 years ago, the first microbe that can actually degrade plastics was found in a PET recycling facility in Japan called Idionella sakaiensis. And from then, the term microbial plastic degradation was born. And it literally means, and I'm just going to start my laser pointer, that we have natural microbes such as bacteria, fungi, and now we even know archaea that produce certain hydrolases, certain plastic degrading enzymes that can then attack certain types of plastics. But we only know 3 types of plastics that are actually naturally attacked so far, which is polyethylene terephthalate with about 120 petases, so PET degrading enzymes known. We know about 20 polyamides, and that's quite new research that was actually only released in the last 12 months, which is really exciting. And then we also know about 30 polyurethane degrading enzymes. But unfortunately, natural plastic degradation is very, very slow, and it won't make a significant difference in the plastic pollution crisis. So researchers such as the Fritz Lab and our team, we want to increase the efficiency of this process. And so we came up with the idea of producing the fastest plastic degrading enzymes with the fastest growing bacterium in the world. And this fastest growing bacterium in the world is called Vibrio natriegens. It's a gram-negative gamma proteobacterium, and it's ubiquitous in marine environments such as seawater, sediment, but it's even a symbiont for some marine organisms. Its most distinct and interesting feature is its extremely fast doubling time of less than 10 minutes in optimal conditions, which is nearly double the speed to like most E. coli strains, and definitely faster than your Streptomyces Tilmann. So yeah, I'm very much in love with the fastest growing organism in the world. And it also grows to higher cell densities, which is obviously very useful for production of natural compounds and proteins. And very recently in our lab, but also in a lot of other labs around the world, we developed a lot of genetic tools to make the bacterium more accessible. And these include plasmid toolkits, CRISPR, and other. So for my PhD project, I'm interested in tackling PET plastic pollution crisis by producing petases with an engineered Vibrio natrigen strain, because Vibrio natrigen is not a natural producer of petases. And then these secreted petases, they hydrolyze the ester bond between the terephthalic acid monomer and the ethylene glycol, first producing M-het, which is the dimer, and then petase and metase hydrolyze the M-het into the 2 separate monomers, which can then be taken up by an engineered Vibrio natrigen strains, fed into the central metabolism used for energy recovery, biomass production, as well as the production of compounds of interest. But this is still a very slow process, even though we use synthetic biology and the fastest organism in the world. And we also now know that we cannot simply recycle our way out of the plastic pollution crisis. So it needs to have a more holistic and global approach. And so as a second option for solutions, we are also interested in coming up with plastic alternatives. And we use PHB, which is short for polyhydroxybutyrate, and that's a natural fat, a bacterial fat that Vibrio natrigens produces to store especially carbon in times of need. And so we feed Vibrio natrigens different carbon sources, and then we make it produce PHB effectively for us. But before I could do all of these great things with a beautiful bacterium, I had to come up with optimized tools to build plasmids and also to do genome engineering. And of course, I haven't been doing that by myself singly. I'm in a big group effort here. And the next couple of slides, I want to talk about how we're actually using synthetic biology tools and how we have been optimizing them to work in Vibrio natrigens. So the first Golden Gate toolbox that was specifically built and tested for Vibrio natrigens was called the Marburg collection in 2018. And it was the idea of this fantastic iGEM team from Marburg in Germany that actually won the grand prize at the iGEM competition in the US with that idea of having a huge library of parts that were specifically tested and built for Vibrio natrigens. And because Vibrio is such a fast-growing bacterium, it is the opportunity to accelerate the design, build, test, and learn cycle for synthetic biology, and it's therefore the optimal environment for part characterization. And it enabled a very systematic, modular, and flexible approach to engineer this non-model organism, which is fantastic. So what the Marburg collection originally offered was level 0 parts with lots of different genetic parts that you need for protein expression. So you had promoter parts, ribosome binding sites, coding sequences, and terminators, as well as origin of replications for the plasmids and different antibiotic resistance cassettes that all had a color code. So they were all encoding either a GFP or an scarlet red fluorescent protein cassette so that you can optically distinguish. And then up to 8 of those parts would be combined into a level 1 unit that would then hold a transcription unit. And then up to 5 transcription unit could then be combined into a level 2 plasmid. And this enabled the very modular and flexible assembly of the plasmid of choice with a huge library of tested parts, more than 191. They also introduced so-called connectors, which sit upstream of the promoter and downstream of the terminator part. And these connector parts just allow a maximum versatility and flexibility, including the flipping of transcription units and the leaving out of transcription units. So we can also jump transcription units and just use, for example, 2 transcription units rather than 5, if that's what the plasmid is supposed to look like. In general, that collection characterized a lot of Anderson promoters for the constitutive expression of a target protein, lots of different ribosomes and terminators. And they also characterized how well Vibrio natriegens was suited for expressing different fluorescent reporter proteins. And what was really interesting to see was that some of the expressed proteins seemed to actually be toxic and cause metabolic burden and stress, because when expressed, the growth rate actually significantly dropped. And that could also be observed when scarlet was produced with very, very strong promoters or ribosome binding sites. And again, the growth rate would drop significantly. And that goes hand in hand with observations that were done in E. coli before, which we can potentially call the bacterial growth laws, that say that if you overexpress an unused protein, it just takes up a chunk of the proteome and then it cannot be allocated anymore to your normal metabolism or to your translation through ribosomes. So you're kind of like blocking the normal proteome from its normal work, and so the growth rate drops. And we see that in Vibrio natriegens. And so the learning from this was that when you want to produce a protein in Vibrio natriegens, you need to be very careful with metabolic burden. And it's best to actually have a growth phase and separate that from production phase if you want to have high titers and an efficient production. So this collection was used for a couple of years. It was used for further genetic tool development in Vibrio, for the expression of recombinant proteins and more basic research. And throughout the years, we actually understood that there's a lot of optimization potential for this collection. And so together with my colleagues and for the first chapter of my PhD, we developed the so-called VINA 2.0 collection, which is an extension of the current collection. And it will be published in the next couple of weeks. And the collection will, for the first time, be available on AdGene. So we're very excited to finally share all of these parts, now more than 250, with the global community. So what we have been working on recently is the introduction of more inducible promoters. We can now also build operons, and we have a tool for part management. We will also introduce a more efficient design for dropouts. We have curated overhangs for less cross and self-ligation. And we now also combine the plasmid toolbox with the anti-CRISPR method, so we have more efficient genome engineering. And I know this was a lot, so I will explain this step by step in the next couple of slides. So let's start with the novel inducible promoters. In the original collection, we only had the PTET and the PTAC, so the ATC and the IPTG depending inducible promoters, and we wanted to increase the number of inducible promoters in Vibrio natriegens that were thoroughly characterized. And so we looked into the paper from the Voigt lab that used the marionette promoters in E. coli, and we characterized those promoters in Vibrio natriegens, and we can see that most of the inducible promoters are actually active in Vibrio. Some are a little bit leaky, so they have a high basal expression, and we kind of find them unsuitable for work in Vibrio nitrogens. And then we also check those promoters for growth phase dependent effects, and we can exclude those for the ones that we think are functional in Vibrio. And then we also looked at the population heterogeneity with flow cytometer and actually showed that except for naringenin as an inducer, which is potentially slightly toxic at high concentrations here, all the other population heterogeneities are quite small. And so all of those promoters actually give quite a homogeneous population, which is nice. One of the most important questions about inducible promoters for us was if we can actually use them in parallel and independently in the same cell. So we tested for orthogonality. What we did, we had a plate reader experiment where we had strains that expressed an scarlet, and that expression was controlled by an inducible promoter. And then we added different combinations of inducers and saw if we actually have crosstalk, so if we get an scarlet signal, even though the non-corresponding inducer is present. And you can see that we get the high signal when the corresponding inducer is present. Then you have the two leaky promoters. And then interestingly enough, you have two inducers that seem to always give a little bit of signal, which is naringenin and OHC14. And both of those chemicals probably muck around with the quorum sensing system and mechanism in Vibrio nitrogen. So there's definitely a lot of research potential to understand what those two compounds do with Vibrio nitrogens and quorum sensing. All right. Then next, we also looked into the possibility to build operons, which we couldn't do before. And together with the help of Renee and Wing, we now can not only combine up to 5 transcription units in a level 2 plasmid, but we can actually build an operon with up to 5 operon units. That means that there's only going to be 1 promoter in the first operon unit and 1 terminator in the last operon unit. And in order to build these operons, we needed to change the connector parts. So in the first operon unit, the connector part would span over the terminator and the connector so that the transcription would not be terminated. Then in the middle operon units, the connectors would span over the promoter and the terminator part. And then the last operon unit would have the terminator to terminate transcription, but would skip the promoter part. And we built an operon of proof of concept with 4 genes that encoded proteins that were necessary for the production of deoxyviolacin, which is a purple dye that you can see with your eye. And then the last operon unit was for the expression of SFGFP, superfolding GFP, so we could measure green fluorescence and get a signal from that. So we built that operon in a level 2 plasmid successfully, about 10 kb, and sequenced it. We got the oxyviolacin production, which you can see here, and you can see the different color in comparison to the wild type. And we also got GFP depending on this amount of present inducer, because we built that whole operon with an inducible PTET promoter. Then we also wanted to improve the dropout design of the collection. And this is actually what people give me the feedback on is one of the most interesting parts of the collection, which is really exciting for me. So imagine you want to build a library, for example, for testing inducible promoters. Traditionally, you would need to build all of the test constructs from 8 parts. But if you use a dropout plasmid, meaning that you use a dropout part that acts like a placeholder, you can actually build that once from 8 parts, and then you only have a 2-part assembly for building all of your different plasmids for example, in inducible promoter tests. And that reduction in parts increases your efficiency by up to 100 times. And so we could show that dropout parts are really, really useful, especially in difficult assemblies. So we offer these dropout parts for the different positions from position 2 to 5 in different colors. So you can actually distinguish between your dropout part and your antibiotic resistance cassette and your final construct. And we now also curated those dropout parts to have an inner BPI1 cut site. So you can have a one-pot reaction with two restriction enzymes, BPI1 and BSA1 or BSMB1, to replace the dropout part and enter the new one. And this new design gives about 50 times higher cloning efficiency in comparison to the original dropout design. So that was also very useful. All right, so that was all the innovations about plasmid-based optimization in the collection. But then we also looked into combining the collection and the standards of the collection with genome engineering with anti-CRISPR to make that process even more efficient. So for those of you who don't know, anti-CRISPR is a method that was developed in a lab in Marburg by Daniel Stuckenberg. And it is a very, very efficient method for genome engineering. So it combines the natural transformation ability of Vibrio natriegens with the uptake of tDNA, the homologous recombination, and then the counter selection with CRISPR. So the way this works is that if you introduce your CRISPR plasmid in the strain that you want to engineer, and that's your first step, and your CRISPR plasmid has the TFOX master regulator system on it, which just means that if you induce that with the addition of IPTG, you make your cell naturally competent. There's also an anti-CRISPR system, which is just like a buffering system to catch away and to silent the leaky expression of the Cas9 so that your cells actually survive until the moment that you want them to die. So this CRISPR system, this CRISPR plasmid is in your strain of interest, and then you induce with IPTG, your cells become naturally competent, and then they can take up the tDNA, which then does homologous recombination in your genome, introduces the mutation that you want, either a knockout or a replacement or an insertion. And then your majority of strains will not be edited, but you can get rid of them by inducing the CRISPR system using ATC, killing all the non-edited cells because the guide RNA targets the original sequence, and you will be only left with your engineered cells. Roland Politan, another PhD candidate in the Fritz lab, he wanted to optimize that method even further. So he found that you could actually use plasmid DNA as tDNA. And that plasmid DNA is much, much easier and faster to build because he used dropout plasmids that already had the homology flanks for a certain number of landing pads in the genome. And so it made the insertion or knockout of those landing pads very, very easy. And then you also build strains where these landing pads have actually an scarlet cassette. So if these non-engineered strains escape the counter selection with CRISPR, they would turn red, and the counter selection would be more easier to spot if it failed. And like I said, he actually showed that Vibrio natriegens can take up circular DNA and do homologous recombination with circular DNA. The method is, or the way it does it is still not described, and we don't really know and understand, but it works and it's good enough for anti-CRISPR to function. And Roland had these, I think it's like 7 or 8 landing packs that he characterized, and we know the phenotype of the knockouts, and they can be used for the insertion of your gene of interest in the genome. So it's a very time-saving design. All right, so with that said, these are all the optimizations of the VINA 2.0 collection that are going to be released in the next couple of weeks. So we're going to be submitting that paper really soon, and we're going to have all of those more than 200 parts available on AdGene. But for me, the tool development is really just a way to actually apply my research for the greater benefit and to tackle real-world problems. So finishing up my first chapter, I then use this toolbox to degrade PET plastics and to also produce PHB. So for the first part of my applied design, I'm looking into producing different petases with Vibrio natriegens. And as a proof of concept, I just very simply use the Ideonella sarcaiensis petase. And you can see that if I overexpress this PETase in Vibrio nitrogens, and then I use BHET as a ester substrate just for a simple esterase test. If I have PETase present, the BHET is mainly hydrolyzed and I get MHET and TPA. Whereas if I have a negative control, most of the BHET is still here. But the problem is that most petases characterized and described so far actually stem from freshwater or soil environments. So they're not really suitable to work with a marine organism in a marine broth. And this is exactly what we're looking to do, simply because working with synthetic biology in ocean water when scaling up would be much more sustainable and save valuable freshwater resources. And in order to tackle that problem, we ventured out and looked for more marine petases in the highly plastic contaminated sediment in Western Australia. So together with the Ocean Institute, we identified these sites. We took sediment samples. We extracted the eDNA, and we made sure before sending it out for metagenome sequencing that it had good enough quality. And when we got the results back, with the help of our collaborators in Hamburg, we built or used the hidden Markov model, which is a sequence-based homology statistical model that looks into the sequence similarity of petases already known and the metagenome data, and then makes predictions. And we could actually predict 104 petases from our sediment from a lot of different types of bacteria, mainly proteobacteria, which was what we expected. And we are currently now testing those different hydrolases and carboxyesterases more specifically for their true plastic degradation potential in the lab. And of course, we use Vibrio nitrogens for the production of those hydrolases. And then lastly, because we know and we learned we cannot simply recycle our way out of the plastic pollution crisis, we also want to use Vibrio nitrogens for the production of alternative plastic materials. And our choice is PHB, simply because Vibrio nitrogens naturally produces PHB, but also because PHB is actually biodegradable even in marine conditions, which is not true and applicable for all so-called biodegradable plastics. So within a couple of days or years, depending on the thickness of your PHB product, this can actually be eaten up by microbes, even in marine conditions. And I'm very excited, I always show this even though it's very small, that we made our first PHB the other day from different carbon sources. And it was very exciting to extract it and have it in my own hands. So with this, I hope that I could demonstrate that we produced a Golden Gate, or we optimized a Golden Gate toolbox for Vibrio nitrogens with more than 250 parts for building operons, for building very efficient dropout plasmids, with a very, very high flexibility of promoters, ribosome binding sites, and terminators. And that collection is actually useful for applying synthetic biology to tackle lots of different environmental problems, such as the plastic pollution crisis. And with this, I just want to thank my entire team and all of my collaborators, of course, including my great supervisor, George Fritz, for helping me out, supporting me, the Forest Research Foundation for being the main funder of my project. And thank you everybody for listening, and I'm very excited to discuss. And of course, thank you to the seminar and both Tisuk and Tilmann today for giving me the stage. Thank you very much.
Fantastic talk. So I really enjoyed. Also, this is relevant to my topic. So let me. Tilmann was there, but suddenly. Oh, there we go. Tilmann, do you have any question or comment? Oh, you are mute right now.
My internet connection just dropped out, so I didn't hear the last sentence you said.
So do you have any comment or the question? for Anna?
I
Okay.
have actually.
Okay. So
But
let me
are
start.
there some questions from? Yeah. So I was wondering. So first, congratulations to this fantastic toolkit. I think this is amazing to have for a non-model organism to have this flexibility to work with. So I really like this and already made a note to look into it if we can get inspired for some of our plasmids. Uh, I'm not a specialist in all this plastic degradation. I was just wondering the example you showed with the PET, this phthalic acid, is this very toxic to the strain or can they cope with the concentrations you will get?
Yes, you nailed it. So I think people traditionally use Pseudomonas and relatives for these very complicated aromatic compound utilizations because it's just very inherently able to deal with all the radicals that are produced and flying around. And Vibrio is a bit of a princess. It's very quick, but it's also sometimes a bit fragile when it comes to metabolic burden, but also the aromatic compounds. So we have actually done adaptive evolution, and I'm currently analyzing the mutations, and it's really promising. Like I see mutations that are there in all strains, and that suggest that there is very specific adaptation to these conditions. So that's really, really exciting, and that's also hopefully going to be published this year. But yeah, so that's definitely a question that Vibrio nitrogens, I think we haven't really understood yet how and which metabolic compounds it's good to use and which ones not, and which ones we potentially need to be a bit careful with. Yeah.
Okay. Tilman, do you have any other comment or question?
And then I just have a very naive question. Whenever I hear these plastic-eating bacteria, are there any plans how to contain them to the areas where you want them? If I just think about all the new cars that are full with plastic,
Yes. So
and of course,
like I think that's probably also TSOC's expertise, but we're both working in that. Currently, they are very, very, very slow. I would say to the point that I'm not even sure if they're ever going to be good enough for industrial application. So it's really a playground at the moment to just understand how good we can get them in the first place. But of course, like if we ever should theoretically get to the point where they're so good that they would be a danger to infrastructure, then it would be like with other organisms. You know, we have PC4 labs in the world because we do research on pathogens, and that's very useful because we understand how to treat diseases, but we also understand how to contain them. So I think it would be probably similar to something like that. But at the
again,
moment,
I wouldn't at all be afraid to use it in a biotechnological process. I think there is so much experience to not let your organisms out. It would be more if you really go large scale for the oceans, you would need to set
like
the
our research
more organism free.
is more
And then, of
it's
course,
more
this
for
becomes way
contained
more.
biotechnology.
Yeah.
Sorry, I didn't want to talk over you.
Sorry.
Tilman, do you have any other thing?
I think these were the main ones.
Okay, so we have some question from audience. First, Victoria asked question. Thank you so much for the fantastic talk. As a fellow microbial plastic degradation researcher with a much slower growing strain, your work with the VNet is incredible, promising for contributing to solving the plastic crisis. I'm particularly interested in your research extending the marble collection for operon construction. So question is, can you comment on the limits of these operons in terms of number of genes, also the relation of order to express, uh, such as do you find that genes in the first position of operon tend to be expressed more strongly? Or were there any unexpected findings there?
Yes. Yeah, very good question. So the design of the marble collection allows a maximum of 5 operon units in one operon, just simply the way that the overhangs and the connectors are designed. You can always go for fewer, but you cannot go for more in one operon unless you completely restructure and redesign the collection. We haven't tested about the strength of expression for the different positions in the operon, but we could show that GFP, which was an independent operon unit, so it didn't have to work together with the other operon units to have a signal. And we put that on purpose on the last operon unit, and we got a signal there. So we can definitely express all 5 operon units with the signal that we can measure. And the reason we think that we didn't get a very strong deoxyribolase in color, because normally that's like dark purple rather than like this grayish that we saw, it's probably more because of the stoichiometry between the operon units, and that's probably something you would solve with the ribosome binding sites and the strength of those to alter. But yeah, we could definitely express all 5 operon units with one inducible promoter, and we got a signal for everything.
Okay, another question from Matt Pavlovich. He's the chief editor of Trend in Biotechnology at Cell Press. The question is, what's the intended downstream application of PHB? It is a popular thing to make from waste streams, but I'm curious how much industrial demand there is for it. Do you know if any manufacturer make commercial products out of the PHB?
Yeah. So currently, so-called biodegradable plastics are less than 1% of the global market. But I think with the sustainability aspect really taking over economy slowly, it's going to grow significantly. There are quite a lot of startups, not only in Australia, but worldwide, that actually produce PHB. There's actually one in Perth as well. They make it from seaweed. And I think the demand is probably bigger or will be bigger than the actual capacity of producing it. And I think it has the potential, especially with applications where you don't need any long-lasting plastics. So like single-use plastics for packaging, where it can definitely replace petroleum-based plastics quite easily. And these startups already have products that are made from PHB. So that's something very easy to Google. Yeah.
So to add to that answer, PHA, the similar polymer actually produced by the one company called CJ. That's a Korean company. I believe they kind of done some contract with one US company before, and then they actually commercialized those product. One caveat would be the market demand, because they need to find the real market, and then they kind of already struggling in terms of the selling those products. But
Yeah.
they made those products out of, I believe, glucose, not the waste.
Yes. And I think another problem is that at the moment, these technologies are still more expensive, so they're not yet price competitive. So that's definitely something that will need to change.
Yeah. Hopefully, I mean, in the future that change. Let me double check whether we have more questions. Okay. I have multiple questions, but I see the time is already 11. Let me just quickly ask one question. You show the data with the enzyme, and I believe you tested that on in like 30 degree or some temperature, room
Yes, that's only
temperature.
37.
37 for that enzyme degradation in the ocean salt condition or the normal media.
That was actually lysis buffer, but we also have tests for just ocean medium.
So ocean media
So
typically higher salt, right, than the
yes.
normal media. Got it.
Yeah.
Okay. That's the exciting. Okay, so now I see the time is a little bit more than 11 Central Time. Let's close, and then we have the informal chat afterward. Okay, so thank you all for joining and staying today. We'll meet again on January 30th. Oh my God, already almost end of the January. The Thursday, the same time, the same Zoom link. We'll have Professor Vincent Norex at the University of Minnesota and Professor Sujun Ju at Texas A&M University. As usual, the follow-up informal chat will occur without recording. Please stay here. If you are interested in chatting with us, I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. 1 second.